Hi, everyone. My name is Jessica Giordano, and I'm really grateful for the opportunity to speak to you today, along with Dr. Wapner, about the intersection of prenatal genomics and newborn screening. Today, we're going to focus on three main things. We're going to describe the current status of the expanding and evolving fields of prenatal genomics. We'll touch on some recent advances in fetal therapy, and then we'll really focus in and hope we have time afterwards to discuss some opportunities for how the prenatal genomics field and newborn screening stakeholders are able to collaborate. So given the audience, we thought it would be a good place to start actually in the NICU. So we're aware of how advances in neonatal genetics really informs even our own practice. So some recent data in 2017, where 278 critically ill newborns were sequenced, found that more than a third of those individuals were found to have a molecular diagnosis. And interestingly, a lot of those had an atypical or unrecognized presentation of the genetic disease. And also, very importantly, over half of those cases showed that there was a redirection in medical management based on that diagnosis. So a redirection of care, an initiation of a new subspecialist, and so on. But we've been paying careful attention to that and have noticed that a lot of the diagnoses that are seen in the NICU actually overlap with the diagnoses that we see in prenatal exomes, specifically in the setting of congenital anomalies. And I'll get into more details about that today. Certainly advances in technology has improved diagnostics in the postnatal space, but also in the prenatal space. The advent of karyotyping in the 1950s and 60s brought on the ability to do amniocentesis. Dr. Wapner led the charge in having microarray become a standard practice in the last decade. And we've also been now doing next generation sequencing through Exome and Genome in a standard way in our practices. So there's been progress in both the invasive side with CVS and amnio, but also all of that technology is going to be used to harness the power of non-invasive methods as well, which we'll talk about today. So I wanted to start off with a case of someone that I actually saw personally, of a 35-year-old woman who had her first pregnancy naturally conceived with di twins. And you can see in the left picture of the ultrasound the clearly clubbed foot, and you can also see in the right picture that the arm is in a fixed position and the hand is very clearly clenched. So those were the findings in both of those twins, and this family elected to pursue exome sequencing, and we actually found that each fetus was compound heterozygous for pathogenic variants in the rhabsin gene, and both parents were carriers. And this particular condition is associated with a congenital myasthenic syndrome, and we know that 1/3 of rhabsin variants actually have arthrogryposis, as seen in our case. And this became very important because having a specific genetic diagnosis in the setting of myasthenia syndrome really indicates which medications one should choose. And also, it's also important because other medications can actually cause harm. So having the specific etiology is really important in direction of care. So the treatment was guided by the genetic results prenatally. And I'd also note that getting this information prenatally really decreased the time of the diagnostic odyssey for this family, which is also an important piece. What we're really seeing is also a paradigm shift for prenatal diagnosis. And as our technologies keep expanding and we're able to find a larger number of phenotypes, pregnancy termination is only one of the many uses of information obtained prenatally. And to highlight that further, I wanted to note that benefits of sequencing prenatally are varied. So that includes improving pregnancy management, selection of a different delivery site, changing the type of delivery, selecting in utero management and different treatments. We'll touch on some of those later. As I just mentioned, it also can reduce patient uncertainty and anxiety during pregnancy. It improves the perinatal transition, and in very severe cases can allow for a clear plan for compassionate care. And of course, it also maximizes reproductive options in the future. So I'm going to talk about a couple of the different technologies that we're using in our prenatal practices. And I'll start off with prenatal exomes in the setting of structural anomalies. So as many know in this group, structural anomalies impact about 3% of births in the United States and are a leading cause of infant death at about 20%. And our evolving technologies through the years have found a cause in many cases, including about a quarter to a third are caused by karyotype abnormalities, and then 5 to 7% are due to microarray deletions and duplications. But as our genetic technologies expand, we're actually finding an increasing number are genetic. So I have some of our data from our own internal exome study, where we sequenced about almost 500 trios who all had a variety of different types of anomalies. And we found that 58.6% in this group had a de novo dominant etiology to their congenital anomaly. A little more than a quarter had a recessive etiology, and the remainder had an X-linked recessive. Unsurprisingly, we get a positive finding more frequently when there are multiple anomalies. So 26.6% in the setting of multiple anomalies, and then about 8.5% when there's a single anomaly. Also notably, there are a number of genes that do recur across our cases. So there were 11 genes in 25 cases, and that's helpful. And you may see some of these genes are very familiar and identified in a lot of NICU and postnatal studies. But we also found that there were 44 unique genes in our cohort. So many of these genes are not represented in some common screening paradigms, and really would only get picked up through this sort of more expanded exome or genome approach. Also notably, from the data coming out of our colleagues' work in the UK, there's many new prenatal phenotypes being identified, and in their cohort, they identified that 11 diagnoses were never seen prenatally. So there was no data out there to support that those conditions would have really been suspected. Additionally, we noticed that our positive rate changed over time, and there's a couple of reasons that could explain this. I think our expanding knowledge of the prenatal phenotypes has allowed us to reclassify some variants, and we pay attention to the literature to do that. Also, the medical literature expands and finds more candidate genes that are now associated with human disease, so we try to keep track of that and reclassify things. And then also follow-up postnatally allows us to identify children that are really growing into a phenotype. So we may find something prenatally that we don't have enough data to support is causing a disorder, but when we follow up postnatally, the child sort of grows into that, and then we can be sure that it's the cause. So our positive rate has gone up over time. It has been relatively stable at this point, somewhere between 14 and 15%. Through our experience, we've noticed that there are some unique challenges in the prenatal sequencing interpretation space. There's really incomplete phenotypic fetal data due to the limitations of ultrasound and just the temporal limits that are inherent to fetal development. So we're working with a really limited phenotypic picture. And then on the other side, there's a lack of data in the existing databases about the prenatal phenotypes, which could be because some of the more severe phenotypes are lethal or cause a fetal demise, and they're therefore never tested or even documented in a database. We've also noticed that in some cases, there may even be different inheritance models in prenatal cases versus what is seen in childhood or adulthood cases. So the data from this population is currently informing the development of some non-invasive screening techniques, which is always something that is sort of the big goal, to try to get all this information for families in a non-invasive way. One of the other technologies that's seen a huge growth in the last decade has been carrier screening. And to remind everyone, for many years, clinicians have offered a gene-by-gene approach to carrier screening, where patients and couples who are considering pregnancy or are pregnant pursue testing for a handful of recessive and X-linked diseases. That's been classically ethnicity focused, and then cystic fibrosis was really the first model for a pan-ethnic molecular carrier screening test back in 2005. All these changes in next generation sequencing has allowed us to screen for many more disorders with a far higher detection rate, a quick turnaround time, and lower costs, which has been a phenomenal change in our practice. We're working very hard to emphasize with our GYN patients that the best time for screening is really in the preconception period, so families can get information ahead of time and potentially pursue in vitro fertilization and pre-implantation genetic testing. But also we do see that many people do testing early on in pregnancy, which can be helpful to inform their pregnancy management. So I wanted to highlight some data from a JAMA paper that Dr. Wapner was an author on in 2016, where they modeled the fetal risk of genetic diseases that were identified by expanded carrier screening. So in this study, they looked at over almost 350,000 individuals and reviewed their carrier status for 94 severe or profound conditions, and specifically looked at the variants that were definitely pathogenic. And they presented their data in terms of affected pregnancies. And interestingly, 1.5 to 2% of couples were both carriers for a recessive disease. And overall, about 1 in 550 pregnancies in the US are predicted to be affected with a severe or profound recessive condition. Also notably, current ACOG and ACMG guidelines are predicted to miss over 50% of affected pregnancies across every ethnic group. And that data is, the frequency of these diseases is highlighted on the chart in the right. So in thinking about those conditions, I think it's helpful to give the full context of that. And that 1 in 550 number is really higher than a lot of the numbers for the conditions that we do commonly screen for now. You know, for many decades, we focused on screening for Down syndrome. It has an incidence of 1 in 800, ONTDs with a 1 in 1,000, and even cystic fibrosis has an incidence of 1 in 3,400. But collectively, all of these recessive disorders that we were studying in this paper account for 1 in 550 pregnancies. And again, they do represent severe and profound genetic disease. It's just that each one of them is individually rare, but collectively they really add up as an important public health issue. We did look at the data from our own center and found that we had about 9,000 patients screened, and there were 70 carrier couples or X-linked recessive carrier moms. And of all of those listed out on the right, only one condition would have reliably presented with fetal anomalies. So these conditions on this list are really not highly represented in the ultrasound anomaly differential diagnoses. So expanded carrier screening is filling a different sort of gap. It's targeting Mendelian disorders that do not present with ultrasound anomalies, such as Fragile X, hemoglobinopathies, a variety of storage disorders. And they're also not fully represented on newborn screening panels. Some of these conditions are on newborn screening panels, but certainly not all of them. And since we are New York based, I did specifically look back to compare our carrier screening panel with what is available in New York State, and found that about 19% of conditions screened by our standard expanded screen are also covered by New York State newborn screening. So newborn screening and expanded carrier screening are really complementary sort of tests at this point in time, and represent just a difference in timing for when these conditions are being looked at. So we would ideally like to screen everyone well before pregnancy, but there are a variety of reasons that that doesn't happen. And certainly people can do their carrier screening while they're pregnant. And also, as noted earlier, a number of conditions that are causing fetal anomalies are due to de novo variants, which cannot be detected before pregnancy. So we've been putting a lot of effort into leveraging all these advances in the diagnostic space to doing these things in a non-invasive sort of way. Certainly, that would be a patient preference. So there's really two methods that are primarily being looked at right now. One is looking at shedding fragments of placental DNA that really circulate in the maternal circulation, and the other is detecting whole fetal cells that are circulating in the maternal circulation. In terms of detecting de novo disease, it is becoming a more and more important problem because we're seeing higher rates of advancing paternal age in the population, and we know that de novo disease happens more often with advancing paternal age. And as noted earlier, 2/3 of the fetal anomaly cases are solved by a de novo variant. And this chart here is showing data where we compared the de novo cases in our own cohort to a currently available commercial test that does look for de novo variants. And the way that technology is set up now, only up to a third of our cases could have been picked up with that commercially available test. But we do hope that we can make further advances and all of these cases could eventually be picked up in a non-invasive sort of way. So I'm going to pass it over to Dr. Ronald Wapner to talk about advances in fetal therapies and some of our collaborative ideas.
Thank you, Jessica. Certainly, the vast majority of individuals listening to us today are certainly aware of the dramatic ability that's occurred over the past 3 to 5 years in treating of neonatal genetic disease. And here's just an illustration of probably one of the most well-known and most important advances, and that's the ability to screen spinal muscular atrophy. That's the ability to treat spinal muscular atrophy in the very early newborn period. And spinal muscular atrophy, as many of you know, can occur in a number of different formats, but one of the most common is the severe infantile form, in which the children have basically no tone whatsoever and are significantly compromised. Although this has really been a very, very severe diagnosis in the past, we now know that using neonatal genetic therapy with oligonucleotides, with anti-oligos, and sometimes with gene therapies, can dramatically improve the disease. Many of us know that most recently, with the presence and treatment with anti-oligos, the treatment of this disorder very early in the course, even in the neonatal period, has dramatically improved the outlook, with children that may have died previously now able to walk with minimal residual problems. And this was begun in 2016 with Spinraza. So the question becomes whether or not we can do the same form of genetic treatment, not after someone is born, but early in the prenatal period. And with spinal muscular atrophy, this is important because even if treated at birth, these children will still have some residua. And although the earlier the treated, the better, there's very good evidence that the disease process and the atrophy of the nerves has already begun in utero. And the question has evolved, should we or can we be able to do genetic treatment during the fetal period to dramatically decrease the long-term consequences? The next slide illustrates the fact that there are advances in fetal therapy. And I just point out a article from Science Technology that basically summarizes the field, and it's entitled "The Fix is in Utero." I think that one has to understand, because there's much fear about genetic treatment and gene editing in particular. And in this particular case, we're not talking about germ cell editing, in which any changes can be transmitted to future generations. We're really talking about somatic cell gene editing, in which the cells in particular organs or the cells in the individual can have genes edited, but will have no impact on the germ cells and the future. Why do I think this is important? Because there are lots of advantages, if one thinks about it, in treating these conditions during the fetal period as opposed to the newborn period. And those of you who are pediatricians know that there are treatments with bone marrow transplants and with enzymes that have been demonstrated to make a difference. But where we have failed significantly has been with many of the most severe diseases, like the neurologic form of Gaucher's. And the reason is that the damage has either occurred in utero, or the damage is in the central nervous system, which in general is not able to be reached during postnatal treatment. But if you think about it, during development, there are many ways to overcome these deficits. First of all, the fetus has an incredibly immature immune system. So that would minimize the formation of the immune response of the individual to the vectors that are being used for the gene therapy, or the new proteins that are being formed once the gene itself has been entered. So that really we can develop during pregnancy immune tolerance, so that even if later in life you need to do additional treatments, the fetus would be tolerant. Second of all, the cells are more actively dividing during development. And certainly one of the tricks to successful gene therapy is to get it into as many cells as possible, because an individual that's treated is kind of a mosaic. Some cells have the gene therapy involved or occurring in those cells, and others do not. But when you have a very actively dividing cells during development, you really have a greater chance of both introducing the vectors and infecting the cells that you want to be treated. Third of all, the fetus can, in the middle of pregnancy, be very small, so that doses are smaller. But I think the most important point to keep in mind is early in pregnancy, and maybe even through the late second trimester, the blood-brain barrier is completely or partially permeable. So that if you need to treat things that are in the CNS after birth, you would have to give direct treatment to the CNS, which has not been very successful. Alternatively, if used during the fetal period, it will be able to have the treatment cross the blood-brain barrier and successfully treat the CNS complications. Now, all of this obviously goes back to our ability to diagnose many of these genetic conditions in utero at a time that may be ideal for treating an individual. Much of this has been demonstrated to be feasible, and in animal studies, all the way from mice through monkeys. And there are a few studies now in early stages doing this for certain genetic diseases. So this is no longer kind of a fantasy. This is a field that is moving forward. So I hope that we have pointed out that the world has changed. With advances starting with ultrasound, moving through genetic changes, etc., we can diagnose many, if not the majority, of genetic conditions in utero. And many of these are either not identified on newborn screening, or perhaps maybe better to be identified earlier than in the newborn period. So prenatal genetics is really informing our ability to do these things, although, as all of you can imagine, there certainly continue to be technical and socio-ethical issues that tell us that we have to be very, very careful how we introduce these technologies and how they're used. For instance, a real discussion has to take place of what conditions we should be screening for. What are our guidelines? It used to be early in the development of screening, our guideline was it had to be a lethal disease, one that had severe cognitive disabilities, or one that would have to be treated immediately at birth. Well, we now believe that it's time that we re-envision what that should be, and certainly that's a discussion in our field that continues to be taking place. The other problem is, as Jessica pointed out clearly, that interpreting these findings is still in its infancy. And we need to, as we move from exome sequencing to genome sequencing, we are really, really learning about a lot of things that not only had we previously not understood, we still don't understand. So there's a world of work still to be done. Next. In order to accomplish this, we formed the Fetal Sequencing Consortium. And part of the reason that we are talking to you today is that Amy and her group in Screen have been incredibly inviting and collaborative in helping us develop a registry in which all of the sites in the US are contributing their fetal sequencing information so we can begin to have much better reliability in predicting the phenotypes of many of these variants and also decreasing the number of variants of unknown significance. Part of what really we need to talk about, and I believe this is the greatest forum to do this, is the transition from a fetus to a neonate is totally artificial. And I think we still have to start thinking of our patients being the neonatologist patients, the neonatal patients being our patients. And from the standpoint of genetic disorders, much of what we are seeing are much closer to what you are seeing in the newborn periods and the nursery than they are, and we've learned this as we've sequenced fetuses, than they are with what the pediatricians are seeing later in life. So hopefully this discussion is just the beginning of how we can leverage our combined knowledge to not only identify these, but to help answer some of the important developmental questions, ultimately leading to treatment. So for that reason, I hope that we will continue to start putting our minds together, putting our data together, and putting our experience together, and begin to understand this field a lot better. So our world's changing, your world is changing, but I really think the collaborative discussions of how we adapt to these changes and what we can learn from the new tools is going to be critical to the future. So again, I just really want to thank the Amy and the group for even inviting us to do this, and hopefully we will be seeing a lot of you and talking and working with you. Thank you very much.